My name is, good afternoon, guys. My name is Gulfaraz Khan. I'm Professor of Viral Pathology at the College of Medicine, UAE University, which is based in the beautiful city of Al Ain in UAE. My interests are primarily on infectious diseases and pathology. I'd like to introduce our next speaker, Dr. Cybel Mehawej. Dr. Cybel is an assistant professor at the Department of Human Genetics in the Lebanese American University. She got her PhD from Paris Descartes University and then went to Boston for a 2-year immunogenetics training at Boston Children's Hospital. It's a beautiful place. I was there a few years back, Cybel, or many years back rather, at Harvard Medical School, which is part of the medical school. And she basically focused, while she was there, she focused on identification of molecular basis of primary immune deficiencies. Today, she will talk to us about precision medicine in diseases of immune dysregulation. Dr. Cybel, the floor is yours.
Thank you, Dr. Khan, for this introduction. I would also like to thank the Center for Arab Genomics Studies for organizing this interesting meeting. Can I share my screen?
Go ahead, yeah.
Can you see my screen?
We certainly can, yes, thank you.
Okay, so in the coming session, I will be discussing briefly precision medicine in the field of primary immunodeficiency disease and in particular in immune dysregulation disorders. Primary immunodeficiency disease, or PIDs, are a heterogeneous group of more than 400 different entities that affect the development and/or the functioning of the immune system. Patients with PIDs suffer from recurrent or opportunistic infections that occur in addition to several other manifestations such as allergies, failure to thrive, lymphoproliferation, autoimmunity, and autoinflammation. PIDs have been considered for a long time as rare disease. However, increasing evidence suggests that the real prevalence has been underestimated. And in parallel, the development in the field of immunogenetics enabled the identification of several new entities and the implementation of novel therapeutic approaches, such as personalized therapies or targeted therapies. So what is precision medicine? As was very nicely presented in yesterday's module, precision medicine is an emerging concept in the field of medicine that consists of tailoring therapeutic approaches based on the genomics and/or the proteomic profile of a patient. And those approaches present several advantages over the standard therapies because in many cases, one size cannot fit all. Now, personalized medicine is the fruit of several years of research in the field of genetics. Indeed, the implementation of next-generation sequencing approaches allowed the identification of several new genes, among which many are involved in PIDs. Based on the latest classification from the International Union of Immunological Societies that was published in 2019, 416 different PID entities are recognized. They are divided into 10 different categories, and there were more than 430 genes that are linked to these PIDs. And as of today, I can say that there are definitely more than 420 or 450 different PID entities. Now, the identification of the genes involved in this disease allowed also the elucidation of the molecular mechanisms driving many of these entities, and also the introduction of novel concepts in the field of immunogenetics. Indeed, and since their first discovery or recognition in the 20th century, immunodeficiency diseases were long considered as diseases that are due to an underactive immune system. such as the case of severe combined immunodeficiencies or the X-linked A-gamma globulinemia, etc. And they were considered as separate entities from the autoimmune disease that were basically recognized as disease characterized by the presence of autoantibodies, or basically antibodies targeting self-proteins or self-antigens. However, with the advances in the field and the recognition of several new entities, there were different entities that were discovered, and they associated the increased susceptibility to infection with also autoimmune manifestations or immune dysregulation manifestations. So nowadays, autoimmune manifestations and, let's say, immunodeficiencies or increased susceptibility to disease, to infections, can co-occur together. And this is why the term inborn errors of immunity was introduced to combine all those PIDs. And today we consider autoimmune disease not only the disease restricted to the presence of autoantibodies, but mainly all diseases that are due to a defective immunological tolerance, and they can manifest as with the presence of autoantibodies or with an overactive immune system. And all these concepts were very important for the establishment of therapeutic approaches. And in addition to the concept that the new paradigm of inborn errors of immunity, and in addition to the loss of function mutations that have been long recognized, the concept of gain of function mutations associated with several PIDs and also in particular immune dysregulation disorders or autoimmune disorders has become very well recognized. All this paved the way towards the implementation of targeted therapies. Now, in PIDs, targeted therapies are especially effective against immune dysregulation and autoimmune disease. And this is what we're gonna see in the coming few minutes. I will start with the first example of immune dysregulation, which is activated phosphoinositide kinase syndrome, or APDS. APDS is a combined immunodeficiency. So this disease affects both the humoral and the cellular immunity. It is characterized by T-cell senescence, immune deficiency, autoimmunity, lymphoproliferation, and increased risk for lymphoma, such as EBV-driven lymphoma. The onset of the disease is typically in childhood with sinopulmonary infections that often lead to bronchiectasis over time. And recurrent or persistent infections due to herpes viridae can also be seen frequently in patients with APDS. Now, APDS 1 and 2 are caused by mutations in the PIK3CD or the PIK3R1 genes, which both basically modulate the PI3K-AKT signaling pathway, and we're going to discuss this. So the PIK3CD gene encodes the P110 delta catalytic subunit of a kinase known as PI3 kinase, while the PIK3R1 gene encodes the P85 alpha regulatory subunit of this kinase. So basically, this is like an inhibitory subunit, while this is a catalytic subunit. And this PI3K kinase catalyzes the phosphorylation of the PIP2 to PIP3, leading to the activation of downstream signaling pathway. We have to remember that this leads to the activation of the AKT-mTOR signaling pathway. In patients with APDS, we can see either a gain of function in the gene encoding the catalytic subunit, so more activity of this kinase, or loss of function mutations in the gene encoding the regulatory subunit, and thus less of this negative regulation, leading both to increased activation of this downstream signaling pathway, which is the mTOR. Now that the mechanism driving this disease is clear, we can understand the rationale behind the therapeutic approaches that were developed. Indeed, the first approach is the use of mTOR inhibitors, such as rapamycin, that inhibits the mTOR signaling pathway. And in fact, mTOR's inhibitors were shown to improve the immune phenotype of patients with APDS, and treatment has been found to decrease hepatosplenomegaly and lymphadenopathy, to restore T cell proliferation, and to treat non-neoplastic lymphoproliferation. However, I have to mention that the response can be less satisfactory for cytopenias and gastrointestinal symptoms. Another treatment for APDS is basically some selective PI3 kinase delta inhibitors, because we need to downregulate this pathway. So 2 registered trials were done to establish the safety and efficacy of these drugs, one based on oral administration of a drug while the other is inhaled, and the results showed that the drugs were well tolerated and they led to the reduction of lymphadenopathy and splenomegaly and the improvement of cytopenias. Also here, we have to always bear with the side effects, and a long-term use of those mTOR inhibitors or PI3K delta inhibitors in those patients might be critical because of the possible effect on the genomic instability in B cells. That's why in several cases, in several patients with primary immunodeficiencies, a hematopoietic stem cell transplantation is at some point needed, depending on the entity, of course. The second example of disease are CTLA4 haploinsufficiency and LRBA deficiency. And I'm going to discuss both together because we're going to see that both share the same molecular mechanism. So those diseases are immune dysregulation disorders characterized by humoral immunodeficiency and autoimmunity, including autoimmune lymphoproliferation. Patients present with recurrent sinopulmonary and viral infections and with significant infiltrative autoimmunity affecting at least one non-lymphoid organ, such as the lungs, the gastrointestinal tract, and the nervous system, in addition to autoimmune cytopenias. Now, what's the genetics behind those diseases? Is that patients with those diseases have either a heterozygous loss of function mutations in CTLA4, which encodes a receptor, cell surface receptor, that we're going to discuss in a bit, and this leads to CTLA4 haploinsufficiency with autoimmune infiltration, or CHAI, or in the case of LRBA deficiency, we have homozygous also loss of function mutations in the gene encoding and LRBA, leading to LRBA deficiency with autoantibodies, regulatory T cell defects, autoimmune infiltration, and enteropathy, or LATAIE disease. Now, for the sake of simplicity, I'm going to use the term CTLA4 and LRBA deficiency in the next slides. Now, why both diseases share the same molecular mechanisms? This is because LRBA is basically a member of a gene family involved in vesicle trafficking and in the endocytosis of ligand-activated receptors. While CTLA4, that is a receptor expressed on the cell membrane, undergoes endocytosis, where it is either degraded in lysosomes or recycled back to plasma membranes. How is the recycling done? Thanks to LRBA. So in other words, LRBA plays the role of a chaperone for CTLA4, enabling the recycling of this molecule and preventing its degradation in lysosomes. So when we have a loss of LRBA or homozygous loss of function of LRBA, this will lead to rapid shuttling of CTLA4 vesicles to lysosomes, and CTLA4 will be degraded, and a loss of LRBA will also lead to a loss of CTLA4. Now, why is CTLA4 important, and why losing CTLA4 expression leads to autoimmunity? This is because CTLA4 is a crucial T cell inhibitory receptor. So it is expressed on activated T cells. So after their activation, the T cells express CTLA4 on their cell surface in order to maintain the homeostasis and to avoid their overactivation. And as a receptor, CTLA4 will compete with another receptor expressed on T cell surface, which is the CD28, that is very important for the co-activation of T cells. So, in other words, when CTLA4 is expressed on the surface, it will compete with CD28 for the same ligands. And while the binding between CD28 and CD80 will lead to the activation of T cells, the binding between CTLA4 and the CD80 or CD86 will lead to an inhibitory receptor. So it's a kind of competition with the co-activating receptor of T cells. Now, I have to also mention that I said that CTLA4 is expressed on T cells after their activation, and it is constitutively expressed on a subset of T cells known as regulatory T cells or suppressive T cells. And the aim of those T cell subset is basically to inhibit or to suppress the activation of the immune system. This is why using CTLA4 will lead to the loss of regulation, leading to an overactivation of the immune system. So either we have a loss of CTLA4 or a loss of LRBA, that is the chaperone of CTLA4. Both will lead to a loss of CTLA4 and thus to autoimmune manifestations. That's why targeted therapies have been developed, and they consist in the use of abatacept or belatacept, which are CTLA4 immunoglobulin fusion drug. What does this mean? We have a drug that comprises two domains that resembles an immunoglobulin or an antibody, let's say, structure. It has a common fraction of a subset of immunoglobulins, which are the IgG1, while the extracellular domain resembles CTLA4. And the common chain has been modified to avoid the activation of the complement system or the ADCC, which is a kind of cytotoxicity. So the aim of this drug is basically to use the extracellular domain to mimic the presence of the CTLA4. And in this case, when we use this drug, there will be a competition with the CD28 and a sort of repression of the T cells. So in a normal scenario, after their activation, or here we're showing the regulatory T cells, they express the CTLA4 on their surface, and thus they are able to inhibit the overactivation of other T cells, such as the T follicular helper that we're showing here. In patients with a with the CTLA4 or an LRBA deficiency, we don't have much of the CTLA4 expression, and this will lead to the loss of the inhibition, leading to an overactivation of the T cells. Now, when we use the treatment in those patients, we are trying to mimic the presence of CTLA4 by giving the patients those CTLA4 immunoglobulin fusion drugs. They are acting as if the CTLA4 is expressed on Tregs, and they are basically suppressing the overactivation of the immune system. Now, several trials have shown the efficacy of those therapies in patients with CTLA4 and also in patients with LRBA deficiency, because both share the same pathway or mechanism as I mentioned. And the treatment showed to reverse cytopenia, to reverse interstitial lung disease, and to improve the lung function of patients. In addition, also to the reversion of some autoimmune manifestations, such as here the diabetes. Now, those therapies are very promising. However, we have to always consider the limits of those therapies. And in the case of abatacept and belatacept, there is increased susceptibility to infections, especially viral, and to malignancy because of their immunosuppressive activity. And this limits the use of those drugs currently for a long period of time. And here again, we have to consider probably in those patients for a long term, other also therapeutic strategies. Another targeted therapy that can also be used in the case of LRBA is basically trials were done to use chloroquine or hydroxychloroquine, which is a lysosomal inhibitor drug. So this drug inhibits the lysozyme. So in patients who have loss of LRBA, usually the CTLA4 goes to lysosomes where it gets degraded. By inhibiting the lysosomes, we are basically trials, in vivo trials showed that this leads to the increase in CTLA4 expression in vitro in the regulatory T cells and a better outcome. However, in vivo trials are still showing some controversial results and probably would need more time and more functional validation, more in vivo studies. Now, the third and last example of disease is the example of STAT1 and STAT3 gain-of-function, disease linked to STAT1 and STAT3 gain-of-function mutations. What are STATs? STAT, or signal transducer and activator of transcription, are a group of transcription factors. They are expressed by immune and non-immune cells. They are crucial in regulating immune and inflammatory responses. Those STATs act downstream of the receptors of several cytokines or growth factors. What happens in a actually in a cell is that the binding of a ligand to the cytokine will lead to the recruitment of a type of kinases intracellularly known as Janus kinases or JAKs. Those will be recruited to the intracellular or intracytoplasmic tail of the receptor. And because they are kinases, they will be able to phosphorylate this tail. And once the tail is phosphorylated, this will enable the binding of STATs to this intracellular phosphorylated tail or domain. And once STATs are binding to this domain, they will get phosphorylated by themselves, and their phosphorylation will enable their dimerization. And once we have STAT dimers, those will be able to translocate into inside the nucleus and act as transcription factors and regulate several other genes. Now, there are different types of STATs, and these act downstream of several receptors. I'm going to discuss STAT1 and STAT3. STAT1 act downstream of the receptors of interferons type 1 and 2, IL-10, IL-2, IL-21, in addition to several others. While STAT3 acts also downstream of type 1, type 2, type 3 interferons, receptors, IL-6 receptor, IL-10, and IL-21. Now, the interesting part related to those genes is that in this case, in the case of STAT, we have one gene that can be linked to several different clinical phenotypes. How is that? Because if we have a loss of function mutation, we would have a phenotype, while when we have a gain of function mutation in the same gene, we would have a different outcome. Here, for example, with the STAT3 gene, loss of function mutation, dominant negative germline mutations, for example, are associated with autosomal dominant hyper-IgE syndrome that is characterized by eczema, skin abscesses, and recurrent pneumonia, while heterozygous STAT3 gain of function mutations lead to early onset autoimmunity, including type 1 diabetes in infancy. In this talk, because we're focusing on immune dysregulation, we're going to discuss the disease linked to STAT1 and STAT3 gain of function mutations. In fact, STAT1 gain of function mutation leads to a disease characterized by chronic mucocutaneous candidiasis. And patients with STAT1 gain of function mutations present with recurrent bacterial, viral infections in addition to fungal infections, and with in 30% of the cases with immune dysregulation.
Dr. Cybel? I think there seems to be some connection problem. I don't know whether she's, we lost connection with her or not.
She did say that if this happens, she will try and log in from her 3G. So let's give her like 10, 15 seconds. That's all it should take.
Okay.
Yeah. Yeah, I think she's back. Cybel, are you back? Yeah. You're muted, Cybel. No.
Can you hear me now?
Yes, we can.
Yes, excellent.
I'm
Welcome
sorry,
back.
we have some internet issues because of the weather. Where should I continue with the stats?
You still have about 5 minutes, Cybel, so if you want to just...
Okay. Where did it interrupt?
You were on the STAT signaling pathway, so...
Okay. So basically, we discussed the STAT signaling pathway, and we said that those stats, can you see my screen or not yet?
No, we can't. We cannot.
Okay. Now you should be able to see my screen.
Yes, we can. Yes, excellent. Yeah.
Okay. So if we were on this slide.
Now you were on the next, I think you were on slide 30, if I remember right.
Okay, perfect. So with the diseases.
Yes.
So we're going to discuss STAT1 and STAT3 gain of function, or the disease linked to STAT1 and STAT3 gain of function mutations. You can't hear me.
Yes.
Now you can?
We can hear you.
Okay, I'm sorry. Okay, so gain of function mutations in STAT1 lead to chronic mucocutaneous candidiasis, and patients present with recurrent infections, including bacterial, viral, or fungal, in addition to autoimmune manifestation or immune dysregulation manifestation, such as hypothyroidism, type 1 diabetes, autoimmune cytopenia, and SLE. They also present with cerebral aneurysm and vasculopathy frequently, in addition to malignancy. While STAT3 gain of function is basically an early onset autoimmune and lymphoproliferative disease that includes lymphadenopathy and splenomegaly. And patients can present with autoimmune manifestations, mostly enteropathy and cytopenias, and in addition to non-malignant lymphoproliferation, including lymphadenopathy and splenomegaly, and an interstitial pneumonia and recurrent infections, in addition to acquired short stature, which is a peculiar feature of the disease. Now, because we explained that in the case of a gain of function of STAT, we would have an increased STAT signaling. So this is why JAK inhibitors, or inhibiting the kinases that act upstream of the STAT, has been basically used as an approach for treating patients with STAT1 and STAT3 gain of function mutations. Now we have to know that there are several JAK inhibitors, and the use can depend on the availability of the drug, on the toxicity profile, in addition to the disease that needs to be treated, etc. And the use of JAK inhibitors was shown to be very efficient and successful in patients with STAT1 and STAT3 gain of function mutations. And this therapy basically led to the improvement of cytopenias, interstitial lung disease, and total parenteral nutrition dependent enteropathy in those patients. Patients also improved their ability to control infections to a certain extent, and candidiasis was responsive to the treatment. However, again, we have to bear with the limits, because therapy with JAK inhibitors may cause elevation in liver enzymes. That's why this needs to be controlled, in addition to a probability of a mild thrombocytopenia in the patients, and also to the possibility of viremias that can occur, especially when JAK inhibitors are combined with other immunosuppressive treatments. We might also see a herpes reactivation in the patients. That's why they might need to add a prophylaxis in addition to the JAK inhibitors. Now, in conclusion, we have shown that the improved understanding of the pathogenic mechanisms of several newly described PIDs led to the implementation of targeted therapeutic strategies. And the novel mechanism-based approach is known as precision medicine. It is basically a powerful and effective approach in reversing clinical manifestations of immune dysregulation in many cases of PIDs. However, limitations still exist, and these approaches can be used to ameliorate the clinical condition of patients prior to a definitive therapy, which can be the hematopoietic stem cell transplantation. Here are my references, and thank you for attending.